Hey students, Professor Gideon here. We're going to look at the results of the bacteria lab now. I put this into a PowerPoint format and I want to talk to you about what has happened in the last 24 hours as our bacteria has grown in the Petri dish. So take a look at this time lapse. This is showing us growth on a Petri dish. Now this Petri dish contains sheep's blood. That's why it's actually red. But this would be how it looks before the bacteria and fungi begin to grow on it. And then by time lapse, we begin to see these very small little dots form on the Petri dish. And you can actually see this without a microscope. You can see this with the naked eye. And we see that these different shapes, now we call these colonies, are different colors and textures and sizes. And that's going to be important to how we actually evaluate the plates that I did in the experiment from yesterday. So let's take a look at those plates. So if you'd open your books to page 152, we'll take a look at the chart there, question 11. Here it's asking us to record our data from this experiment, and you're going to be recording the data that I collected. So remember, the first plate I inoculated was fingerprints after I touched the bottom of my shoe. You can see there's quite a lot of growth as a result of me touching the bottom of my shoe. And so what we want to do is we want to observe number of species and number of colonies. Now, first of all, let's define what we mean by colony. So a colony is defined as a visible mass of microorganisms that originated from a single cell. So recall when I transferred the bacteria from my hands, we couldn't see those single cells, but when we transfer them onto this nutrient source, this source of nutrients that we call the agar plate, when we transfer them on there, we provide an opportunity for these single-celled organisms to divide. And they divide and divide and divide many, many times to create billions of copies of themselves. And then they begin to form this visible mass, okay? And so we want to determine from the chart the number of colonies. So how many individual spots do we see? Realizing that some of these are very large. And then we also want to describe the number of species that we see. So species, when we're looking at the species, this is where you're looking at different colors, textures, or sizes of colonies. Because each species of bacteria or fungi will produce a unique shape on the plate, okay? So we see that we've got this big jagged edged guy, right? He is one colony actually, but huge. That's a huge one. And we see that there's several others, like here's another one of the jagged edge colonies, okay? And probably one over here as well, and one over here as well. That's a particular species. Okay, here's another one. I see one over here, jagged edged. Okay. Then we see a lot of these little white ones. So that would be another species, second species. So this is species 1, this is species 2. Then, let's see, this one, this is sort of interesting, sort of several layers to this one. I would say that is a third species. And then we have some really large ones that are more rounded on the edges. I see another one over here as well. I'd go with a fourth species on that one. And then we also see some that are maybe a little lighter colored. let's go with a 5th species on that one. You know, this is an approximation, so don't feel like you have to use a magnifying glass and try to see every little detail on my Petri dish. So do the best you can, but we would record this as the number of species. We'll go ahead and say 5 in our chart. Now, number of colonies. Wow, there's quite a lot there. We'd want to count each individual spot on here. Okay, and you can approximate that. Some of the plates you're going to see, there's a grouping of some of the colonies, like over here is a grouping where they sort of merge together. And the reason why they merge together is that as they grow, as the cells divide, if the original cells, the original sort of colonist, okay, that original bacteria that was on my hands, if it was close to another colonist or another bacterial cell, when they started to divide, they sort of merge. They sort of merge together. Anyway, so that makes it challenging to actually count individual colonies, and the dirty plate is pretty dirty. There's definitely a lot of bacteria. Remember, I just touched the bottom of my shoe, and I told you, yeah, there's a lot of dirty bacteria in there. Anyways, I'm going to say greater than 100 conservatively there. Here in this box, you can describe what you observe in the plate, okay? And so maybe the shapes of the colonies. So we see small white colonies. We see large rough colonies. We see smooth, large, etc. So that's just giving us some descriptive words we can use to describe our Petri dish. Now we also want to go ahead and record results for the water plate, the soap plate, and the 2 objects. And why not? Let's go ahead and add one more box because we did hand sanitizer, if you recall. So write in hand sanitizer. Let's add that one because we had 5 plates. Okay, so I'm not going to do all the plates for you, but I do have the pictures in this PowerPoint, and I'll post this PowerPoint for you so you can go through and, oops, sorry about that. You can go through and actually count the colonies that you see. Here's our water plate. We see several colonies, not any of those big jagged edge ones though. Those guys are gone. Lots of the smooth, round, beige, sort of white colonies. We see a lot of those. Soap plates looking better, okay, compared to water only. Hand sanitizer is not looking so good on this plate. And then here is the objects. No colonies on the cell phone. Maybe one, actually. Looks like we may have one here. One lone guy here. Surprisingly clean for a cell phone surface. And then here's the ear. You saw me swab my ear. We get quite a lot of colonies in there, and actually so much growth here that you can't really count too many of the individuals. So you can record this in your lab notebook. We use a word for this called lawn, and that just means there's a ton of bacteria. And I told you that's actually to be expected. It's not that my ear is particularly gross compared to anyone else's ear. It's just that there's a lot of bacteria on our skin, and that makes a component of the microbiome. We want them there, actually. As long as they're not causing harm or causing ear infection or anything like that, they actually outcompete pathogenic bacteria or disease-causing bacteria that can cause pathology in the ear. So anyways, you've got some plates to observe and record on page 152, question 11. No class data, so you can cross out question 12, and you should be able to find the answers to the remainder of those questions in the lab manual, and I hope that was helpful.